Ever wondered what happens to red blood cells when they're exposed to different concentrations of saline solution? Well, you're in for a treat. We're exploring the intriguing world of osmolarity and lysis in red blood cells. Our hypothesis? Saline concentrations, high or low, will each uniquely impact these tiny life carriers. Will they swell? Will they shrink? Or maybe, just maybe, they'll do something entirely unexpected. Intrigued? Let's dive into the details of this fascinating experiment. To test the hypothesis, a simple experiment is required. So let's dive into the details. Our first step is to prepare various concentrations of saline solution. We'll start with distilled water, which has 0 salt content, and then move on to create solutions with 5, 10, and 15% salt concentration. This range will allow us to observe the effects of different saline environments on red blood cells. Once our saline solutions are ready, we move on to the second step, which involves the extraction of a blood sample. This sample will then be divided into portions. Each portion will be immersed in one of our prepared saline solutions. The third step is the most exciting one. After allowing the blood cells to sit in the saline solutions for a set time, we'll examine them under a microscope. What we're looking for here are changes in the size and shape of the red blood cells. If our hypothesis is correct, we should see the cells behave differently in each solution. In distilled water, we expect the cells to swell and possibly burst due to osmosis. In the 5% solution, the cells should maintain their normal shape. In the 10 and 15% solutions, we anticipate the cells will shrink due to water leaving the cell in an attempt to balance the salt concentration. And there you have it, the experiment is complete. Now, what do these results mean? The results of this experiment might surprise you. Fascinatingly, we discovered that the saline concentration had profound effects on the red blood cells. In high saline concentrations, a process called crenation occurred. The cells began to contract and became dehydrated as the water within them migrated out to the surrounding solution. This was an attempt to equilibrate the concentration of salts. It's important to note that saline concentration plays a significant role in the osmolarity of cells. Osmolarity is the measure of solute concentration, defined as the number of solute particles per liter of solution. It's a factor in how water moves across cell membranes. In contrast, when the red blood cells were exposed to environments with low saline concentrations, they started swell and even burst at times. This phenomenon is due to water flowing into the cells in an attempt to balance the lower salt concentration. The influx of water caused them to expand and, in some instances, rupture. This process is known as lysis. But the most intriguing observation was made in isotonic solutions, where the saline concentration was the same inside and outside the cell. In such conditions, the cells maintained their regular, healthy shape, neither shriveling nor swelling. So, to sum up, the saline concentration has a remarkable influence on the osmolarity and lysis of red blood cells. Isn't biology fascinating? So, what's the big deal about this experiment? Well, it's all about understanding osmolarity and lysis, two vital concepts in biology and medicine. They're key to things like IV fluid administration, where getting the saline concentration right can mean the difference between treatment success and failure. This experiment is a stepping stone to understanding these intricate processes. With this knowledge, you're one step closer to understanding the complex world of cells. Until next time, stay curious and keep exploring.